ask me questions. And then number 3, I listen to all of you guys and the things that you talk about with Friedreich's ataxia to understand life with FA that is not always captured in textbooks or publications, so that we can make sure that we're paying attention to that with everything that we're doing at Reata.
Thanks, Kara. Mark?
I'm Mark Payne. I'm a pediatric cardiologist at Indiana University School of Medicine. So that's my full-time job, and my part-time job is to work for Chondrial Therapeutics. So I'm the Chief Science Officer for Chondrial Therapeutics. And what we work on at Chondrial is protein replacement therapeutics to deliver the missing protein into where it needs to be inside the cell. So I've been working on Friedreich's ataxia since about 2003. I run a basic lab at IU, and I also do a lot of clinical trials at IU. I do still see pediatric cardiology patients. It's only 1 day a week. Happily, I don't have to be on call anymore, and I don't have to be in the ICU anymore, so that's 2 thumbs up for that one. I'm really happy about that. But I've been working in this field for a long time, and I'm excited to be at the point where Chondrial is now doing phase 1 trials for protein replacement therapy.
Mark's being modest. He's the Chief Scientific Officer for Chondrial, but he was also the person who discovered protein replacement for FA in your academic lab early on. So, Dr. Milner?
Hi, can everybody hear me? My name is Peter Milner. I'm a basic scientist and physician trained at major medical centers in the US. I was a professor of medicine and molecular biology at Washington University in St. Louis and then Stanford. Went off into the industry 25 years ago, founded a successful billion-dollar company, CV Therapeutics, Aris Therapeutics, where I invented and developed drugs which are now on the market. About 5 years ago, I joined the board of Retrotope, which is a company in Los Altos, so it was a small company. I recognized the advantage of their technology. And then as Chief Medical Officer in the last 3 years, I've hired a very capable team who are sitting over there in the corner. And we are now in phase 3 for multiple neurodegenerative diseases, and I'll explain later how our drug works, how it works in Friedreich's, the results we have, and why we believe it has the potential to be first-line therapy for the big 4 serious neurodegenerative diseases: Friedreich's, ALS, PSP, and Huntington's.
So, Peter, how about if you started to get down the track of telling us a little bit about the drug. So can you tell us what kinds of drugs and therapies Retrotope's developing, how you know that those might be helpful in FA, and can you give us a sense of sort of the level of evidence, right, that's needed or was needed early on in testing those drugs and knowing when to bring them to a clinical trial and kind of a little bit of the history there. And I let the panelists give me a few slides each so that would hopefully help you and help them in explaining a little bit about each of these therapeutic interventions and how they're similar and different from each other.
Thank you, Jen. I'll just give you 5 slides over about 3 or 4 minutes that describe what we do. So right now, our drug is a deuterated polyunsaturated fatty acid, bioavailable for absorption, distributes into the brain and other tissues. We can get to 40 to 60% of all brain lipids are replaced by our non-oxidizable polyunsaturated fatty acid. We've had close to 100 people on it for 50 years, and we've had 19 children on it for a year, one of them on it for 3 years, and no adverse events. We had 1 serious adverse event in the pre-drugs trial, which was diarrhea or steatorrhea, which was limiting. So in essence, we have a safe drug. We have 2 pivotal trials going on right now, one in an ultra-rare orphan called INAD. This affects children. It's a regressive, what's called physical and mental retardation, just like adrenoleukodystrophy. We have commenced enrollment, and we've got about 20 in our Friedreich's ataxia study, the major sites that was shown by Sue Perlman earlier. In May, we're starting a 100-patient progressive supranuclear palsy. This is the disease that affected Dudley Moore, and Linda Ronstadt has it. It's a midbrain tauopathy. We've reversed 3 patients with that over a year. And then in ALS, working with Sue and with Mass General, we halved the rate of progression of 18 patients over 6 months. So by the end of 2021, we should have 4 randomized placebo-controlled pivotal trials. The ALS study is 240, will be done ex-US. So what we do basically is, if you look, it was mentioned earlier about ferroptosis. So I'll explain in a minute in one slide how our drug works. But if you take control cells and you measure mitochondrial function, and then you add iron, as we heard, iron is highly toxic and induces what's called ferroptosis, which is lipid peroxidation. As a result of that, you get mitochondrial cell death and reactive oxygen species. If you add our drug, as you can see on the right, you completely block the effect of free iron. We've evolved many large proteins that cage iron: hemoglobin, haptoglobin, frataxin, transferrin, ferritin. You have to ask yourself, why do we have so many proteins that carry iron? Because you don't want free iron lying around. We use it in cytokines and hemoglobin to carry oxygen, or for redox reactions to generate ATP. So free iron is toxic. We prevent the toxicity of free iron. So we did a phase 2 randomized placebo-controlled study a few years ago that Sue was very helpful in. We treated 19 patients with our drug versus placebo in a blinded fashion. It was a 1-month study, and we saw a 28% improvement in exercise tolerance using cardiopulmonary exercise testing. The p-value was significant. So with that, we're now moving on to a large 60-patient randomized placebo-controlled study in free drug ataxia. Our primary endpoint for that study is this endpoint, cardiopulmonary exercise testing. I've used that before, and the FDA allowed it for hypertrophic cardiomyopathy. It's an integration of neural, skeletal muscle, and cardiac muscle function. Secondary will be the Friedreich's ataxia rating scale, but that's relatively slowly progressive. In PSP and ALS, we have scales that recognize change within a year. So how our drug works is very simple. If you look at a normal healthy patient, they have a normal G for frataxin. They have adequate levels of frataxin, and frataxin shuttles the iron in and out of the mitochondria. So the level of iron, the level of frataxin is normal, and the level of iron is low. And so you don't get much lipid peroxidation. We all have it, normal healthy people. And then you have normal mitochondria. In the disease Friedreich's ataxia, we heard, you get these intronic repeats upstream of the frataxin gene. The level of frataxin is down. It's a normal protein, but the level is low. There are some mutations that actually result in a mixed heterozygote pattern where there's a point mutation as well as normal levels. But the iron accumulates, and the iron sets up ferroptosis, which kills the mitochondria. So we heard that this morning. Our drug is very simple. You still have the intronic expansions. You still have low levels of frataxin. So we're not going to tackle some of the other things that frataxin does. And gene therapy may come along and cure that. You still have the high iron levels. But by giving our drug and getting up to 40 or 60% of all membranes are non-oxidizable, you prevent the lipid peroxidation of ferroptosis and have normal mitochondrial function, resulting in the exercise tolerance I showed you. So in simple, we're not curing the disease, but just like in progressive supranuclear palsy, Huntington's, PSP, ALS, we're preventing progression of those diseases and reversing some of them. So we'll be first-line therapy, hoping in 2 years for a whole range of diseases, and then people will be amenable to definitive therapies, whether it's gene therapy, antisense oligo, or some other therapy that cures them. But meanwhile, they'll be on our drug, which is a gel cap that's taken 6 tablets a day, well tolerated, safe, been in children, as I said, for 3 years, and you'll block progression, but then allow patients to have definitive curative therapies later.
Farah, if I can ask you to go next, I'll bring up your slides for you. I couldn't figure out how to get every company's slides into one presentation because you all have like really fancy settings. Yeah, so I had to kind of go about this the old-fashioned way. There was crazy things that happened when I tried to combine the presentation.
I'll go ahead and stand up so I can see all you guys over here too. Like I said already, I am not a scientist or a researcher. I'm not quite as much of an expert as Peter and Mark are, but I'm going to try my best to explain a little bit of what's going on with Reata and what we've been working on. So for the last several years, we've been working on a clinical trial that we call the MOXIE study. It's using a drug that was originally known as RTA-408. Then we came up with the really fun name of omaveloxolone, and we decided that was a little too hard to say, so I'm just going to call it OMAV. So earlier this morning, Dr. Perlman talked a little bit about how mitochondria in Friedreich's ataxia don't work as well as they're supposed to. And Liz mentioned the same thing towards the end of her presentation. So that's kind of where we think that our drug, OMAV, can take its place and help out a little bit. So I borrowed a slide from our scientists, and this bar graph on the left-hand side shows FA cells in a petri dish, and it measures the mitochondrial function, most simply. On the left-hand side, the black bar is healthy cells that don't have FA, and it shows where their mitochondrial function is at. The middle gray bar shows cells of FA patients, and where the mitochondrial function is at for them. You can see that it's lower than the healthy cells. And then the blue bar shows when OMAV is added to FA cells, and you can see the mitochondrial function goes back up. So that's kind of the rationale for what made us think, okay, this drug might actually be able to make a difference in Friedreich's ataxia. So we developed a two-part clinical trial. That's where the MOXIE study is. And in the first part, we tested out all sorts of different dose levels of the drug. It's an oral capsule, it's just taken once a day. And we tried everywhere from as low as 5 milligrams all the way up to 300 milligrams. And we wanted to figure out what's the best dose level that works, or that we're going to see some type of activity taking place, while also making sure that it's safe and not causing any serious side effects that are going to make it so our patients are not going to be able to tolerate this drug. So we saw, A, it seemed to be pretty safe, relatively few adverse events or side effects. And second of all, at the dose that we found to be most effective, we saw that neurological function seemed to improve. And this was based off of a test called the mFARS, where a neurologist will ask you to touch your finger to your nose and then to their finger, or stand on one foot, and they put it together and give you a score. So we saw in the first part of our study that that score improved, which is why we moved into the second part of the study. So just in October, a couple months ago, we made an announcement about the results that we saw from that second part of the study. And this graph shows those results at the very, very top level. We're still going through analysis of all of the results, so just keep in mind this is just the top level. And you can see there's a line that goes down and kind of stays down. It's a blue line. Those are people who were on our drug. And in this test in specific, the lower your score, the better, kind of like golf. You want a lower score. So it's a good thing for that score to go down. The gray line shows people who were on placebo, so like a sugar pill, no actual drug. You can see at first they went down. That's because we know that placebo effect can happen in trials. But eventually it kind of started to go back up. And so at the end of the trial, you can see that the difference between people who were on the drug and people who were on the placebo was different. So that leads us to where we are now. We've completed phase 2, or part 2 of the trial, and we're now still working on analysis of that to go and talk to the FDA about next steps. In the meantime, we're also continuing on an extension that we call phase 3, which means that every single person that's in phase 3 is for sure on active drugs, so that we can see over a really long period of time what we're seeing from the drug and make sure that we're seeing long-term safety, long-term efficacy. So that's kind of where we are at this point in time. Hopefully I did an okay job as a stand-in scientist. So I'll give you guys just this one last little overview slide, because this is what our legal department approved for me to show to you. So I want to make sure I use it, because that is hard to do right now. So we think that based off of the trial that we've seen with OMAP in MOXIE, that we think there's a clinical benefit to patients with FA in our drug from what we've seen so far. We're planning to meet with the FDA and also other regulatory agencies, the EMA in Europe and others, to start talking to them about being able to submit an application to them. And like I said, we're still continuing to evaluate the safety and the efficacy of the drug in the Part 3 extension. So from day one, we've been working really closely with FARA. We'll continue to do that through the rest of all of our involvement in Friedreich's ataxia, and we'll let you guys know as soon as we are able to provide updates. And in the meantime, we'll also be looking at the feasibility of being able to launch early access. That was a lot pumped into like 4 minutes, so hopefully we can unpack a little bit later, but there's an update on where we are now. Thank you, Sarah.
And Mark, I asked you not only to give us an overview of Chondrial's protein replacement strategy, but I also asked you to address one other question, which is, can you explain to us how protein replacement is made, how frataxin is made? Because I know that's been an important part of the process of developing frataxin. And it's one of those things when people ask why things take a long time, it's not a very exciting thing to think about, but I think it's a practical thing that we've learned from that if you can share a little bit of that, it helps us understand why sometimes it takes a long time to get from those initial lab experiments to human trials.
I'd be delighted to, and I really appreciate the opportunity to talk. So before I get started, I was given strict instructions by the moderator, you can have 2 slides. So like most good speakers, I bumped it up by 50%. I had 2 slides. So, now I got to see what I'm actually doing. So, protein replacement therapy is actually not a new invention. It's all of you know about it. Because all of you know somebody who has diabetes who's taking insulin. That's probably one of our first most successful protein replacement therapies, and that's been around for a while. So we talked a bit about what the basic problem is.
That's a sliding answer, but we disabled it.
Smart move. Okay. So anyway, all of you know what the problem is. It's the gene. There's a problem. And this is from my grad student in the lab who made this beautiful drawing for me. If this is the nucleus, here is the gene that's defective in Friedreich's. And instead of producing 10 to 100 molecules of frataxin, over here it's only 91, goes into the mitochondria like it should, but there's not enough of it, so there's defective electron transport chain. And iron sulfur clusters are not manufactured as they should be. And that's important because iron sulfur clusters are used throughout the cell too, so other parts of the cell suffer. The point in protein replacement is, well, if frataxin is the missing protein, can we get it back into the mitochondria where it can do some good? And the answer is yes. The drug is now called CTI-1601, that's Chondrial Therapeutics drug. And this is a drug, and I'll show you the structure in just a second, but the hypothesis is it goes back to the mitochondria, and instead of having low frataxin, high free iron inside the cell with resultant phenotype of ataxia, cardiomyopathy, and as a cardiologist, I'm interested in cardiomyopathies. The ataxia is something that's attached to it. And so I really focus on this. And then in the presence of adequate frataxin, there's a normal cell function. There's rescue or prevention of ataxia. There's rescue and prevention of disease disease state in the heart. So Jen asked me to talk a bit about how it's made, how it comes about, and how we get to where we need to be. So down here is the structure of the molecule, which is the human frataxin gene, right here, in quotes, human frataxin. There's a cell-penetrant peptide at one end. There's a mitochondrial targeting sequence here, which gets it into the mitochondria. And the mitochondria is going to recognize this lipid right here, protease activity lipid, and that makes the operative chunk of the protein inside the mitochondria where it does its job. If you don't do that, this cell-penetrant peptide will move it right back out of the mitochondria and back out of the cell just as quick as you like. So this is a way to trap the protein product inside the mitochondria where it does its business. Along the way in manufacturing this, here's how we do this. This drug is manufactured in E. coli. So that's a time-honored way of making recombinant proteins. So yes, it's a large vat of E. coli that grows a lot of our protein. So we take this and through a series of experimental batches, we work out purification schemes. We use both protein chromatography, we use gels to analyze what we're manufacturing, use our final protein product, that's the stuff we want to make. And then we have to figure out, well, what's the buffer that this goes into, and that's called formulation. So we have to be able to keep that protein soluble so that when we put it into people or animals, it doesn't turn into brick dust, which is frequently a killer for many drugs. It just turns into something that's insoluble. So the formulation is actually very important. And then we scale this up to much larger batches. For example, we're at 300 liters, for example, right now. We'll be moving to 1,500 liters. So these are big batches of E. coli. The conditions and the purification can change. So this is not a quick process to get this going. Once it is established, fantastic. The manufacturing facilities can generate this. This is what they do for a living. They're very, very good at it. Along the way, there are multiple interactions with the FDA. Now, this process is not just unique to us in Chondrial therapeutics. All the other drugs that have been developed here have to go through the same process. They have a development, they have a manufacturing scheme, and then they have to prove to the FDA that they're making what they're making and that it's safe. So along the way, there's lab development of assays that say we made what we claim we made and that it's safe. So we have to remove all of the endotoxins which come out of E. coli. And we have to show that it's active inside of a living system. We have to go through GMP manufacturing. We have to be able to test in multiple animal species to show that it's safe and efficacious. And one of the things that took this drug so long was to move through animals to show that, hey, it worked in the heart, and hey, it works in the nerve system as well, so they walk better. And then following this, it has to go through conditions called fill finish, which to me as a scientist or a clinician, I look at that and say, well, you put it in a bottle and there it goes. But actually it's a science unto itself. How do you put it into a bottle? How do you get it stable so that you can use it at home for a long period of time? All of this sounds very boring to hear about, but it is integral to manufacturing any kind of pharmaceutical. It doesn't matter if it's protein replacement therapy, an orally ingested drug, for example. These are all things that have to be addressed along the way. Once we have gone through this process here, we then have to interact once again with the FDA. We have to take the stuff that we manufactured here and put it at least into 2 animal species. We've been using rats. This could be a mouse too, but it's bigger, so I'm going to call it a rat. And in this case, we're using non-human primates as well. We have to show that the pharmacokinetics are acceptable. We have to show that the pharmacodynamics, that is, its interaction with the living system, is adequate. And we have to show that it's safe in the animal. I put this slide in here for a reason. This is not a quick process. Trust me, I have learned in the worst sort of way. Years I've been working on this since 2003. And I'm delighted to say it's now in phase 1 trials. But it really takes a village to get from basic science lab in an academic university to a patient on the other end. There's advocacy, FARA, and what you guys may not recognize is how much advocacy FARA has done with the FDA, with the NIH, with government regulation to understand rare disease. That's just, it blows me away how much they've done. The government, all of these organizations are involved in moving from a basic science discovery into patients. FDA, NIH, this was a program that supported development early on. Academia, this drug started out at Wake Forest, it's now at Indiana University, which is part of it. It has interfaced with the Children's Hospital of Philadelphia, which is a fantastic place as well. Research. The reason I put this in down here is the following. All of this has to be supported by funding along the way. So, FARA has funded part of this, the NIH has funded part of this, very hard, National Taxi Foundation. If you want to know where the time sink is, it is here. Because the funding has to be renewed on an annual to 4 to 5 year basis. And that takes a lot of time and it slows down drug development. And it's one of the reasons for why industry, for example, can move very quickly. They do so less of a public forum than academics do. But the funding is really the issue that drives this forward. And it takes a long time to figure out how do you get funding to support it, and are you moving in the right direction? So it's a balance of science and funding between the two. And then once it moves to this point, this needs to be picked up, for example, by some form of company that is going to support its development moving forward. It can be large pharma, like Shire Pharmaceuticals or Bristol Myers, or it can be a small venture capital firm. Actually, Deerfield is not small, but Deerfield picked this project up, and it was through their engagement and support that this program moved from being an academic environment into a biotech company, which is very exciting to be involved with. There have been other funding endeavors along the way. This takes a long time, but once you've reached that stage and once you've worked out the conditions, it's amazing how quickly it can move, and science becomes very exciting, which I can't talk to you about. But the phase 1 trials are ongoing right now, for example, for CK-1601. The other thing that it takes, no matter what the drug is that you have to show in an appropriate model, cell culture, animals, that you fixed the problem. It can be biochemically, you prepared it, or you prevented it, and it has to have some kind of physiological or behavioral outcome readout that you can then go back to the company or to investors or whoever is going to fund this moving forward, and prove to them that yes, we did what we said we can to fix the disease. And this takes time. It takes time, presentation, and publication, etc. So it would be fantastic if we could accelerate this project. Any of it. I think artificial intelligence, for example, is one of the ways in which we can do this by looking at gene signatures of therapeutic interventions. But it really is a slow process sometimes, and it just drives you nuts when you're trying to accelerate things. And you see patients in your clinic, and you're saying, man, I wish I could really hurry this up. It's reached the point now where there are multiple trials going on in mitochondrial diseases. Friedreich's ataxia is one of them. I'm really excited to be there. So that's why it takes so long. Or part of the reason for why it takes so long. So that's my story, and I'm happy to answer any questions.
So you heard a little bit about multiple different stages of drug development, phase 1, phase 2, phase 3. And just for folks who might be new to clinical trial lingo, phase 1 is the very first time something goes into human testing. And a lot of times that starts with healthy individuals and testing the drug for safety in healthy individuals. And I think with both the Reata compound and the Retrotope compound, healthy individuals might have been tested first. Definitely.
Free drugs.
Okay. And for some drugs, we can't test them in healthy individuals to start, and so we do have to start in individuals with FA. And so with the phase 1 study for Chondrial, it is literally the first time the drug's going to be in people, and we start with a single dose, and you monitor safety. And then after you meet safety milestones, you come back and you do it again, but now you start testing with multiple doses. And so that's a multiple ascending dose study. And so this clinical trial will have 2 parts, the single dose part followed by a multiple ascending dose part. And it's all evaluating safety and dose that then helps you move forward to phase 2 studies that you heard about, where you're verifying your dose and you're looking for a signal that the drug is now doing what it's supposed to do, and starting to look for hints of what we say is efficacy. And those phase 2 studies can sometimes be extended, sort of in transition. One of the advantages rare diseases have is that we don't always have to go phase 1, phase 2, phase 3. Sometimes we can parlay a phase 2 into a phase 3 study. And so sometimes the terminology can be a little tricky. But the idea is you move from safety, dose, efficacy, and then in placebo-controlled trials show efficacy over a long period of time and continuing to evaluate safety. All right, so before we open it up, let me just check my time. So before we open up to the audience for some additional questions, maybe could you just tell us a little bit about how, during the development of each of your programs, you've interacted with the patient community, and how do those interactions help inform how your programs go forward? So, Kara, I'll let you start off.
You think the patient advocacy person go first?
And the advantage this time, right?
So I had mentioned that Reata has worked really closely with FARA since day 1, and still continues to do so to this day, which is now nearly 5 years later. Some of the ways that we've interacted with the patient community is by coming out to symposiums like this and going to every single one of the Reata events around the country to talk to patients, to talk to parents, to talk to caregivers, to talk to them about the things that are important to them that we may not be thinking about when we're inside of our walls. So I'll give you an example. We were trying to decide if we wanted to use multiple small pills or a couple of big pills to achieve the dose in our trial. When you read about Friedreich's ataxia, you read that there's a lot of difficulty in swallowing that can develop. So we thought, of course, small pills, of course. What other answer is there? But I talked to a few patients who kind of opened our eyes, and they said, no, actually, my vitamins I take are really big. I'd rather be able to handle a larger pill than drop small pills and then get so frustrated that I'm not going to take it, period. So that was some kind of information that came from interacting with the patient community that helped to develop our strategy in how we made our pills moving forward, just as a small example.
Thanks, Peter, do you have an example?
Yes. Apart from the obvious, which they've been extremely helpful in, which is site recruitment, and you saw the sites that we're using, and patient recruitment, FARA was extremely useful in helping us with a real challenge that there is in Friedreich's ataxia with the Food and Drug Administration. So as I looked at these diseases, I figured you need about 1/6 of the actuarial survival of the disease to judge whether you're having an impact. So in ALS, it's 36 months. So I only need 6 months trials. That's real easy. I've done that. We're doing pivotal out there. Progressive supranuclear palsy strikes men and women in their 50s. They live for about 6 years. So I only need 1 year's treatment. And I've done that, and we've reversed it. Friedreich's was much more challenging. It comes on the age of about 14 or 15, and you live till your 40s. So that's a 24-year actuarial survival. So I would need a 4-year clinical trial to judge whether I'm really impacting the FARA scale. Now, it's very nice to see Reata impacted it in a year. We impacted it in a month, but I looked at this, and when I did the power calculations, I needed, and the FDA asked us to do 3,500 people with free drugs ataxia in the study in 1 year to judge whether we're having effect. That was ridiculous. And Janet Woodcock had said to me, because I do the performance review for the Food and Drug Administration for the United States House of Representatives, every year she said, if anybody in the orphan division or any division asks you with an orphan drug to do a number that is unreal, call me. So I called Janet, and she said, that's ridiculous. So we could not use the FARA scale as our primary. What we needed to do was to use the cardiopulmonary exercise testing, which I showed you the results on. We had very nice results. I could power a study of 60 people. Without FARA support, we had to go to the FDA. I had to drag the cardiorenal people over to Billy Dunn in neurology and get him, who Billy Dunn had never heard of cardiopulmonary exercise testing, which is used widely in cardiovascular drug approval for cardiomyopathies. Finally, the neurology division listened, but it was really thanks to Jan and FARA that we were able to get that change. So now we have a viable primary, and a viable, which is a quality of life, in fact, and a viable study design.
All right. I'm going to open it up because I want to give you all a chance for a few questions.
For the benefit of the viewers at home that will be watching the digital movies and also people at the corners, please could you repeat the question before you answer the question?
Sure thing.
Thank you. Appreciate that.
That was an easy one. Okay. Yes.
Who was it in the House of Representatives that you guys had to go through? Is it a committee that you went through and contacted for the orphan drugs to get this?
No, we went straight to the FDA.
Oh.
Yeah.
But the deputy commissioner, the head of CEDA, Janet Woodcock, had said, in the orphan drug legislation, there are several things that are unique. One is you can get away with a single pivotal trial for approval. 80% of orphans have a single pivotal trial. So you get the orphan designation. Other larger diseases need two confirmatory, two large pivotals. So we went through Janet, and Janet, is the boss of Billy Dunn, is the head of neurology. They're really very good, don't misunderstand me. Somebody down there had asked for 3,500 patients, but when it got up to Janet's desk, who's the head of CEDA, this is ridiculous, you can't do that. There's 5,000 people in the US and 15,000 worldwide. So rational reason and logic came to force because the FARA scale is so slowly progressive, you need 4 years of treatment to really see a statistical significance. It was great for us. When Reata did that, I changed the endpoints at the last minute before we started our study. I bumped it up to our first secondary because now I knew it was validated.
Very good.
Yes.
So I was curious, there's been a lot of buzz about OMA. I've even had family members see something on CBS Business and inquire. So I want to understand once this kind of news, and it's so exciting, we all want our hands on it. So now that you're approaching the FDA, do you know how long that takes? Is there anything we can do? Can we storm the FDA?
Yeah.
So that's probably the number one question that I get from people. And I'll repeat the question first. I should know better with Paul. So the question essentially was, okay, now that we have news about OMEV, what's next? What's the timeline? And is there anything that the patient community can do, including storming the FDA, which I would probably not recommend. I like the idea and I love the enthusiasm. So unfortunately, I'm not really able to give a very specific timeline on what is next. The whole process of drug development follows a series of steps, and if you laid it out on paper and mapped it out on a calendar, in theory, you could lay out dates. But we really have to take it one step at a time. So right now, what we can say and what I'm allowed to say and all that they've actually told me is that we are analyzing our information. We are preparing for discussions with the FDA to talk about submitting an application. I know that's not the answer that you want, but I hope that we can give updates in the future. You as the patient community, what you can do is continue to stay engaged, stay involved, coming to symposiums like this, joining the registry, updating your information in the registry. Every single bit of that makes a difference for us, but also for all of the other drug companies that are working on Friedreich's ataxia. So being here is already step one of what you can do. Maybe not necessarily directly for ONAF with the FDA, but for drug development as a whole in Friedreich's ataxia.
Thank you.
Maybe I'll take a crack at the first part of your question, which is what is the significance of the result? And from FARA's perspective, so the primary outcome measure was this Friedreich's ataxia rating scale. And so for those of you who don't participate in the natural history study, you might not know what this is, but it's a neurologic exam where we try and quantitate how well you can do certain coordination tasks, standing, sitting, finger to nose type testing, things like that. And it's a very standard exam that's done at every single visit. And through the natural history study, where we've been measuring this Friedreich's ataxia rating scale over 18 years now, we know how that scale changes over time. We know how neurologic function in FA changes over time. And so over time, your score on the Friedreich's ataxia rating scale goes up. And what the study showed was that, and Kara showed this on the slide, where the people in the placebo started to have the increase in score over 1 year that we normally see, about 2 points increase each year. The scale goes from 0 to 93, and we see about a 2-point increase each year. And what we saw with the drug treatment is that there was an improvement in the score, a decrease in the score rather than the score going up. And so that's a significant thing, right? Because it's going in the opposite direction, so that's good. But it also helps us inform future drug development. And so when we're using this scale in future clinical trials, kind of to what Peter was saying, how many people would you need, how long would you need to treat to be able to see this difference between placebo and a drug effect? And is it measurable, and can it be statistically significant? And so those are some of the other kind of key aha takeaways that have been really important with the outcome of the result.
Thank you.
Yep. Steve?
So we've heard that there probably, or one of the approaches would be a cocktail approach to the therapies. Where in the, I guess the process when this is prescribed, before it's prescribed, do we understand that the therapies play nicely with one another?
So the question is around a cocktail therapy, and when we are having one therapy that's approved and then the next one's coming on, how do these approaches get tested in combination? Is that?
Yeah, so we've got 3 wonderful solutions in progress that we just heard about. So at some point they'll all be available. Do we know that you can use all 3, maybe 2 out of the 3 at one combo? How does that happen?
Well, I'm going to defer to the 2 cardiologists on the panel who've had lots of experience, I bet, over the years.
While he's answering, could you pop up my slide deck again? There's one slide that outlines it.
Mark, maybe you can start to.
I'll take a stab at that. The first thing to be done is to prove that the compound had an effect. The second thing to prove is will they be compatible with each other? So you can either, for example, mix them together and give them at the same time, or you may give one drug and another, so you alternate drugs, for example. Before you get to that point, what is probably most helpful is to do this in an animal model. And we actually do have good animal models for all 3 of these approaches that we're talking about today. And with the animal model, you can ask some pretty basic questions that you can't ask in people, and get a pretty good heads up both for dose, because the dose might need to change, it may need to drop, it may need to go up. And you can also address the issue of what would you look for I think with the animal models, and this is also kudos to FARA once again, because they have been the ones who have pushed the animal model development. We have pretty good animal models today to test all three of these approaches, and certainly cell culture. And so that can be developed pretty quickly once the, how can I put this? Once the drug companies have decided to sit down and make nice. And if that happens, then things will move very quickly, I think. So the animal models are there, and I think the drugs are understood. So with that in mind, I think it can move quickly. So, Peter?
Yeah, thank you. It's a very good question, and it will be practical. Reality is all in other drugs succeed. What I want to do is show you one patient. This is a patient with late onset Tay-Sachs. This is alpha hexaminidase deficiency. Obviously, it's a different disease from Friedreich's, but they have a very similar phenotype. They have cerebellar atrophy, dorsal column problems. They have an ataxia. They're unable to walk. This lady was 43. She was in a wheelchair. Most of the time, could get out and walk very slowly. We treated her for 70 weeks on our drug. And what you can see, each week, we measured her exercise tolerance by the speed of her walk test. And she did beautifully. Her activities of daily living got better. She was able to get off the commode without help, which she hadn't been able to do. She was able to turn in bed. Turning in bed is a function of extensive function of the elbows. She regained that. Then she decided she wanted, and her family wanted to go into the Genzyme definitive treatment for late onset Tay-Sachs. Great. So we were forced to stop the drug because ours is an experimental drug and that too is an experimental drug. We stopped in October of last year and you see what happened to her in red. Now she's not going to go into the Genzyme drug therapy. She wants back on our drug. When our drug's approved, there's absolutely no reason why protein replacement therapy, gene therapy, antisense oligo can't be given on top of our drug. But you wouldn't want that to happen if you were on first-line primary care therapy that was well tolerated for a whole range of fatal and progressive neurodegenerative diseases. So the answer is, get into the market, get it established in well-tolerated setting. We're not curing the disease, but we're improving function activities of daily living. We may be curing certain diseases like progressive supranuclear palsy, which has a rationale that I could explain offline why it's curative. But certainly with Friedreich's, we're not curing the disease, but we're improving function.
Thank you.
All right, we have time for just one more question. Nick, oh.
Okay. Can I go? My question is for Dr. Payne. I know that Frataxin is currently an injection. Is it possible to do protein replacement in the form, or does it always have to be an injection?
So the question is, Frataxin or 1601 is currently an injectable drug, and the question is, can we come up with a better way to do it? And the answer is, I think, yes. I personally don't want to answer the specifics for how I'm going about that, but yes, I think that we can do this in a better way than getting poked with a needle. That being said, it's a small needle. Diabetics seem to do it because their choices is not great. But right now, the key thing is to just get on the market and get a drug to you so that you can get started reversing the disease. And then we can work out delivery details later. But yeah, I'm already addressing that particular issue. So that's a good question.
Can I just ask one quick question? When you get the okay to put that drug on the market and you throw it into the pharmacies, you got probably a year or so of them trying to figure out how to bill it to the insurances. How do you mainstream that or expedite that? Or besides just giving out maybe possible pharmaceutical coupons, that kind of thing, but is there any other way that you need help to do?
So I'll have to repeat the question.
The question was, once the drug passes approval by the FDA and it moves towards commercialization, how do you work out the details of reimbursement and paying for it? I'm a physician scientist and I got no idea. There are people who understand that, and there are people, and I was discussing this with Dr. Perlman earlier, it takes a whole bunch of money to develop these drugs, and yes, they are commercial companies and they have to get reimbursed, but the real question is, how do you approach the insurance companies to cover the cost of it?
Right.
Insurance companies, for example, are very hard-nosed. They look at the results. If we can reduce the cost of taking care of these patients, then it's, they'll look at that and say, in some fashion, this is how we'll pay for it. Did I answer your question?
You did. The reason I asked is that that's going to be your bottleneck. Is if it's because you do need reimbursement for the medication. That's not the argument. Far from it. It's just, I'm just wondering if there's a system in place where you contact insurance companies, major ones like your Blue Cross, United Healthcare, state providers, ahead of time, and do the pre-auth, because basically pre-authorizations are kind of silly. They're basically a message to the doctor.
Gotcha.
So part of your question is what can be done ahead of time so that
Yes.
there isn't a bottleneck between when a treatment gets approved and patients getting access.
Yeah.
And that's something Reata and FARA are having lots of conversations about right now. And there are, companies have figured this out over and over and over again. And so there actually are pretty set paths to make these things go quickly. One of the things that we're recognizing might be a bottleneck is helping the physicians understand how to prescribe the drug. That the drug exists, how to prescribe it. That's probably a bigger bottleneck than the insurance companies coming up with agreeing to approve the drug. And so we're thinking about how to address greater education to the physician community and what information can be created so that they'll be able to do that in a quick, effective way. But those are all important issues that we are actively thinking about, working on. And you'll be getting lots of survey requests from us for something called disease burden studies. And the reason is we have to document what the burden of the disease is, not just the medical aspects, but the social economic aspects in terms of burden of disease, because that does come back to insurance companies and being able to advocate for their paying for the treatments of these diseases. And so part of the reason we're sending out these surveys and working with different partners to do these economic burden studies is so that we have data that we can share and take to the insurers so that they understand the economic burden of the disease. Paul?
The good news for FA is that this is not an FA question. Every single drug that comes out of the FDA has to go through the same thing. So it's not rocket science. It's just a lot of work, and all of the drug companies should know or can go to people that do know how to make that work as fast as is possible, right?
Exactly. All right.
Excuse me, I have one more question. Will any of these drugs be available? The FDA is a gatekeeper, right? Within the United States?
For the most part.
Yes.
Yes.
Will any of these drugs be available outside?
So the question is can these drugs be available outside of FDA approval or outside the U.S.?
Well, both.
Both.
I'll take a stab at this. So there are different programs that pharmaceutical companies can work on to provide a drug to patients before there is FDA approval. So for example, the phase 3 portion, or the part 3 extension of our study provides drug to patients who participated in part 1 and part 2 of the study. We're also working on what's called an extended access program. It's not in place yet. We still have a lot of work to do to figure out how to make it happen. But this would provide an opportunity to give patients who did not participate in part 1 or part 2 of our clinical trial access to the drug before FDA approval. That's a very simple way to answer it. There's a lot of red tape that has to be worked through before that can actually take place. But it is something that we're working on that is an option.
You guys too?
Yeah, I don't have much
Earlier.
to add except that it's a separate regulatory process in Europe. We've embarked on that. You need an IMPD, MEA approval. They'll use in these rare diseases the data you generate in the US, so they don't necessarily need our ALS study we're doing in Europe just because we don't want to get away of the multiple trials that are going on right now. But we expect our Friedrich's application, once we have the randomized placebo-controlled data next year, will be filed simultaneously in Europe and the US. We've used expanded access to explore other diseases, and the FDA encourages that. Then when we see promising results, we file an IND, we apply for the orphan, and then breakthrough designation. So that's a well-known strategy. Apart from giving it to the disease where it works before it gets to the market, you can use expanded access to explore efficacy in other diseases and mount up safety, good safety data.
Okay, I know we've had some really good conversation. We can come back to it also later today. We'll have more time for questions, but I want to respect that we're a little bit over time and thank our panel for their participation and